Alright, in this video we are going to discuss about the prostaglandins, isoenzymes, antioxidant vitamins and porphyrias, and this video is prepared for the second round of Elsevier. To start with the prostaglandins and other related compounds such as thromboxanes or leukotrienes or lipoxanes are collectively called eicosanoids and these eicosanoids are called eicosanoids because they are derived from 20 carbon compound called prostanoic acid, and since they are derived from prostanoic acid they are also called prostanoids. And prostaglandins were initially found in the secretion of prostate glands. That is why they were called prostaglandins, but now it is proved that each and every cell can release prostaglandins, and that is why these are also called the local hormones. During the synthesis of prostaglandins, suppose they are mainly synthesized from the cell membranes, and they are synthesized in the response of hormones such as epinephrine. And in the cell membrane there is a special enzyme sitting on the cell membrane which is called phospholipase A2. Phospholipase that would lyase the one of the fatty acid, and after the action of phospholipase there is a release of an fatty acid which is called arachidonic acid. Arachidonic acid is derived from essential fatty acid. They are omega-6 fatty acid, and other fatty acids which are essential, they are omega-3 fatty acid, and arachidonic acid is mainly derived from omega-6 fatty acid. They are linoleic acid. So it is very important to take essential fatty acids in the diet. And then these arachidonic acid under the cycle-like enzyme, say suppose this is the cycle, and this is the enzyme is called the cyclooxygenase. Arachidonic acid is converted into the PGG2, and this is converted into PGH2, and this is mainly the precursor for the thromboxanes and prostacyclins, right? These are clinically very much important because it is the balance between, this is very much important to know that it is actually the balance between the thromboxane A2 and prostacyclins which decide the blood clot formation. If the level of thromboxane A2 increases, then the blood would clot because this is platelet aggregator and this is vasodilator. So it is the balance between thromboxane and prostacyclin which would decide the formation of blood clot. We would now discuss the different function of prostaglandins. Obviously, one is a regulation of blood pressure. Some of the prostaglandins such as PGE2 can dilate the vessel, so it can be used as antihypertensive. Next is reproduction. Prostaglandins like PG, E1 and E2 under the name metaprost can be used for MTP and induction of labor. Next function is regulation of gastric secretion. Prostaglandins E2 mainly inhibit adenylate cyclase so that further cascade do not take place. Renal function, it can increase the glomerular filtration rate so that it can increase the urine output. Next is thrombosis. I already said that it is actually the balance between prostacyclin and thromboxane A2 which decides the formation of thrombus in the blood. This was about the prostaglandins. Now we'll describe about the isoenzymes. Isoenzyme, by definition, they are the multiple forms of an enzyme catalyzing the same reaction. This is the only thing which is common in different isoenzyme, different form of one isoenzyme. Otherwise they are different physically as well as chemically, so they'll be different by their physical properties as well as they'll be different by their chemical properties. Now we'll describe the classically one enzyme which is lactate dehydrogenase. It mainly catalyzes the reaction which is conversion of pyruvate into the lactate, and there are 5 different type of lactate dehydrogenase isoenzyme found. They are LDH1, 2, 3, 4 and 5. LDH1 is mainly found in the heart, and LDH5 is mainly found in muscles as well as liver. As I have said, they are only catalyzing the same reaction, but they are different physically as well as chemically. Physical and chemical properties may indicate the pH or km or Vmax value or immunological properties, and everything would be different except they are catalyzing the same reaction. If you want to really know that, these suppose LDH1 is found in the heart, so that it does mean that it would convert pyruvate into the lactate, but in heart we don't want pyruvic acid to convert into the lactic acid, otherwise the heart muscle could get fatigued. So the LDH1 would have low affinity for pyruvate, or it would have high Km value. Similarly, in muscle, the pyruvic acid is gradually but rather rapidly converted into lactic acid, so there is high affinity and there is low Km value. This is how you can differentiate. They are different chemically as well, and these are clinically very important, because normally if you find the electrophoretic property, this is an LDH2 is mainly higher than all of them. This is LDH1, 2, 3, 4 and 5. This is generally higher in the blood, but during myocardial infarction LDH1 rises, right? LDH1 rises. This can suggest after some hours of myocardial infarction that heart has undergone the myocardial infarction. Or in chronic liver disease, the LDH5 level can rise. This is LDH5. This can indicate the liver disease. That is why these enzymes are clinically very important. Next clinically important enzyme is creatine phosphokinase, in short CPK. There are three forms of creatine phosphokinase, CPK1, 2 and 3, and this CPK2 is very much important clinically, because normally it is very much trace in amount in the blood, but during myocardial infarction after some hour at around 20% rise is seen in CPK2, better than lactate dehydrogenase, because this will be not increased if there is muscle hypertrophy or any liver diseases. So it is better than LDH1 marker. This can be used very widely. And if you really want to know other isoenzymes, these are alkaline phosphate and alcohol dehydrogenase. There are several other isoenzymes. We'll not go into the details of them. Next is antioxidant vitamins. Antioxidant vitamins are mainly vitamin E, vitamin C and provitamin A. Their function is to eradicate the free radicals and prevent the damage done by the free radicals to the body. Antioxidant because they would itself get oxidized and prevent oxidation of other structure in the body. Free radicals are mainly produced during normal biological process as well as due to the environmental factors, and can lead to damage of proteins, lipids, carbohydrates and nucleic acid, and this can precipitate several diseases like coronary heart diseases and can lead to cancer. It can promote aging or it can produce diabetes. So free radicals are very dangerous. It is very much important to consume antioxidant vitamins to eradicate the free radicals. Well, look how the free radicals undergo the rapid proliferation and how they are produced, and we'll see how lipid peroxidation takes place. So if you look in the lipid peroxidation, this is fatty acid along with the hydrogen group, and there is normally hydroxyl group found in the cell. So there will be an ion lipid or ion fatty acid which would be formed, and under the aerobic condition this would produce peroxide, and this can react with another fatty acid, can produce hydroperoxide, and these hydroperoxide along with that also one more ion fatty acid is produced, and this chain reaction goes on and more free radicals are produced which are dangerous, right? And chain reaction goes on unless the whole lipid is peroxidized, and there will be no lipid left unless the free radicals are completely eradicated. So it is important for the antioxidant vitamins such as E, C, A. They are called antioxidants because they would prevent the oxidation of this lipid and production of peroxide. That is why they are also called chain breaking lipid peroxidase antioxidants. And to know the free radical stress in the body, there is one marker which is called MDA or malondialdehyde. This is to know the free radical stress, and the clinically it is very important. If the free radical stress is high in the patient, you can recommend more intake of the antioxidant vitamins. Next is very devastating condition called porphyrias. These are clinically very much important, and these are produced due to deficiency of certain enzymes during the hemoglobin synthesis. We'll look at the overview of hemoglobin synthesis to understand porphyrias. This is the overview of the hemoglobin synthesis. Glycine, which is non-essential amino acid which is derived from serine, and succinyl-CoA is an intermediate of the Krebs cycle. And fusion of these two molecules under the enzyme delta aminolevulinate synthase. This is the regulatory enzyme, that is why it is very much important clinically as well, and that would produce delta aminolevulinate. And two molecules of delta aminolevulinate under dehydratase would produce porphobilinogen, and under the enzyme uroporphobilinogen 1 and 3 synthase, that would produce uroporphobilinogen 3. Under decarboxylation, this would produce coproporphobilinogen 3, and under oxidation, this would produce protoporphyrinogen, right? And under oxidation further, this would produce protoporphyrin, and the iron would be added then, and finally under the ferrochelatase, that would produce heme, and under the conjugation of globin, that would produce hemoglobin. This was very basic overview about the hemoglobin synthesis. Now any of the deficiency of these enzymes, say suppose this enzyme or any of these enzyme deficiency, can lead to porphyria. We'll discuss one of the porphyria in detail, that is the acute intermittent porphyria. So this was the overview, and the main deficiency of enzyme is this enzyme uroporphobilinogen 1 synthase. Since this enzyme is deficient, the precursor of, or say prior to this enzyme, the products would be accumulated. So this would be accumulated in the blood, this would be accumulated in the blood, and finally that would be thrown out into the urine, and the color of the urine may change. And these products are harmful to the body as well, because they can produce the vomiting like effect, they can also produce abdominal pain, they can also precipitate conditions which are neuropsychiatric in nature, and they can also produce cardiovascular anomalies. So these are very dangerous products, and to treat this, it is necessary to block this enzyme only, so that further reaction does not take place. So these patients are given hematine that would block the ALA synthase, so there will be blockage of the entire cycle. There are several other porphyrias such as congenital erythropoietic porphyrias or porphyria cutaneous tarda. This porphyria is most commonly occurring porphyria, and this the next is hereditary coproporphyria or variegate porphyria or protoporphyria. The common symptom of these all porphyria is photosensitivity. In acute intermittent porphyria, the photosensitivity is not present because uroporphobilinogen is not present, but these other porphyrias are precipitating conditions which is photosensitive in nature because uroporphobilinogen is produced. This was very basic about the porphyrias, and thanks for watching.